Welcome back to Microbial Concepts. So today's video is related to fermentation technology where we are going to focus on fermentation media. Now fermentation media is a very important part of fermentation process as we require media to grow our culture so that we can get our desired product synthesized or produced by them. Okay, so let's start. So fermentation. So just a quick revision here. So it is a chemical process by which molecules such as glucose or you can say carbohydrates, they are broken down by anaerobic process, okay, or anaerobically. Now anaerobic respiration is a type of cellular respiration where respiration takes place in the absence of oxygen. And aerobic means no requirement of oxygen. Fermentation is an anaerobic pathway which is a common metabolic pathway in majority of prokaryotes and unicellular eukaryotes. Now unicellular eukaryotes like yeast, okay? So this is the cellular level fermentation we are talking, which means which is involved in the metabolism, okay, of a particular bacterial cell or a yeast cell. Now at an industrial level, when we talk about the fermentation, it is a process that is carried out in a large vessel where we grow our microbes, or you can say a culture, microbial culture, in a nutrient media which uh, which we use to produce a economically important product or a byproduct. Okay, so what are the important requirements for fermentation process? First, a fermenter of a proper design, then fermentation media optimized, then microbial culture, you can say a improved strain, because generally to get high output or high yield, strain improvement or um, you can say a genetically engineered strain is also used. Then comes the optimum condition, that is the temperature, pH, okay, and oxygen requirement, etc. So this is about the importance, or you can say the important factors which are very important in case of fermentation technology. Now all these, if they are um, designed properly, then you can get a high yield. Okay, so let's go further towards our today's topic, that is media that we use. Now medium is singular and plural is media. It is a substance which provides nutrients for the growth of microorganisms. Now the nutrients on which the microorganisms they are cultivated, they are generally termed as culture media. Okay, so plural is culture media and singular is culture medium. Now a culture media, it can be liquid, solid or semi-solid, and it is designed to support the growth of microorganisms, and the use of a type of culture media depends on our aim. Okay, so even in fermentation, generally the fermentation is carried out in a liquid media, but even there is solid state fermentation done, okay, where the moisture content is low. So fermentation media must satisfy all the nutritional requirements of a microbial culture. So what are the nutritional requirements? So you can say the fermentation media is categorized into two major and minor components. Okay, two parts, major and minor. So major components are carbon, nitrogen and water, or you can say moisture. And minor components are like inorganic salts, growth factors, vitamins, antifoaming agent, buffers, dissolved oxygen, growth inhibitors, some enzymes and precursors. Now each and every component from minor components, it can differ. Okay, the requirement of such minor component can differ from one culture to another. Okay, you can say that a growth factor is required for one particular culture but not for the other. Okay, so likewise the culture media is designed depending on the product and depending on the culture that we are using. Now depending on the type of microbial culture and type of fermentation used, the requirements of nutrients in the fermentation media, they can change. Okay, so now about the uses of fermentation media. So you can see the first use, you can note it down as um, the one which has the fermentation media, the one which has less nutrients, it is used for the growth of microbes and preparation of inoculum. Okay, not for the actual fermentation process but a part of fermentation process, that is preparation of inoculum. Then the second is where the actual fermentation media which contains large amounts of nutrients and it is used in industries for the production of economically important products. So these are the two uses of fermentation media. And the nutrients which are to be used in fermentation media, they should be formulated to promote the synthesis of target product. That can be either a cell biomass. Okay, that means generally, that means simply you just want the culture to be grown on a large scale. That is a cell biomass or a metabolite or metabolic product. Now about the types of fermentation media. So generally there are two, that is synthetic media and crude media. Now about the synthetic media. So synthetic media, it has a predetermined exact composition of nutrients. Okay, so here the concentration of nutrients can be controlled and redesigned. That means they can be optimized if necessary. Everything is known, how much amount of yeast extract is added, how much amount of carbon source is there, everything is predetermined. So depending on the concentrations, you can increase or decrease a particular ingredient. You can redesign the media if required. So it is used in experiments which are related to, you can that means the first initial experiments before the pilot scale fermentation, where you study the effect of nutrient on the growth and the product yield. Okay, so that is also a part of fermentation process. Then comes the product recovery. So product recovery is easy, as all the nutrients or all the ingredients which are used, they are pure. Okay, so so the product recovery becomes easy. But the disadvantage is the synthetic media is expensive, and thus it cannot be used for large scale industrial fermentation, as the process will not be cost effective. This is about the synthetic media. Now about the crude media. So crude media, it contains rough composition of media. That means it has undefined source of nutrients. Okay, so it can be cost effective, but the recovery of product becomes tricky. And yes, even crude media, it gives you high yield, but the advantage is your process becomes cost effective. Now let's study what are the ingredients of a crude media. So crude media, if you are designing a product on product or the production of a particular desired product on a large scale industrial process, then something you need to design is the fermentation media. So what are the parts that you need to consider? So these are the ingredients. So first is carbon source, then nitrogen, minerals, chelators, vitamins and growth factors, precursors if required, then inducers, elicitors, inhibitors, then oxygen, water and antifoam. Okay, not necessarily that each and every component or every ingredient you will require for your fermentation process, but yes, you need to take them into consideration whether you require this or not. Okay, so let's start with the carbon source first. So the carbon source is required for all biosynthesis leading to reproduction, product formation, cell maintenance. Okay, so carbon source is very important in case of any biosynthetic process, or in case of reproduction, in case of any product formation, and for cell maintenance. So even in most fermentations, it is also used as a energy source. So what are the examples or the options for carbon source to be used in a crude media? So first is molasses. So you can see molasses is the waste waste byproduct, you can say, because molasses has some applications. So I will call it as a byproduct, which is left over waste after um, processing of the sugarcane in sugar industry. Okay, so molasses, then malted barley. Okay, then starch and dextrins, sulfate water liquor, alkanes and alcohols and alkenes, and oils and fats. Okay, so these are the some options for carbon source to be used in crude media. Now what are the factors influencing the carbon source? So as the carbon source is very important and first most important ingredient, it should be cost. You should take in consideration the cost of product, the rate at which it is metabolized, the geographical condition or the location, then what are the government regulation, and cellular yield coefficient. These are the some factors which are or which can influence your carbon source which you are using. Okay, so you need to think about it. You need to consider these factors when you are selecting efficient carbon source. Okay, then comes the nitrogen source. So most industrial microbes, they can utilize both inorganic and organic nitrogen sources. Now inorganic nitrogen may be supplied as ammonium salt, often ammonium sulfate and diammonium hydrogen phosphate or ammonia. And ammonia can also be used to adjust the pH of fermentation. Now, organic sources, they include amino acid, proteins, and urea. Okay, some other options you can use as a nitrogen source are corn strip liquor, then yeast extract, peptones, and soybean meal. Okay, so this is about the nitrogen source, the second most important ingredient from your media. Then comes minerals. Okay, so this is the third most important component, and yes, mostly each and every fermentation media there are some minerals present. Okay, so this is also important. It is a minor component, but yes, it is important. Okay, so all the microorganisms, they require certain elements, not all minerals. Okay, they require certain mineral elements for growth and metabolism. In many media, you will find magnesium, phosphorus, potassium, sulfur, calcium, and chlorine. They are essential components and they must be added. Okay, you will find they are present here and there in your media. Okay, so others such as cobalt, copper, iron, manganese, molybdenum, zinc, they are present in sufficient quantities in the water supplies and as impurities in other media ingredients. Okay, so not necessarily you will always require to add these, they are present in water and as impurities, but yes, they can be used up in your media by the culture. So next is about chelators. So many media cannot be prepared without precipitation during autoclaving. Okay, so hence some chelating agents, they are added to form complexes with metal ions which are gradually utilized by the microorganisms. For example, chelators that can be used are EDTA, citric acid, polyphosphates, etc. It is important to check the concentration of chelators, otherwise it may inhibit the growth. Okay, anything which is excess in your media can harm the growth of your microbes, so you need to check the concentrations. In many media, these are added separately after autoclaving or yeast extract peptone complex. With these yeast extract and peptone complex can form with these metal ions. Okay, then comes vitamins and growth factors. So many bacteria can synthesize all necessary vitamins from basic elements. But for other bacteria like filamentous fungi and yeast, they must be added or they must be given some supplements to the fermentation media. Okay. So most natural carbon and nitrogen sources all also contain at least some of the required vitamins as minor contaminants. Okay, so those can also be used up by your culture. So it depends on the culture which you are using, that whether you require to add a supplement like a growth factor or a vitamin or not, or your culture can synthesize itself. Then comes the precursors. Now precursor is defined as a substance or substances that are added prior to or simultaneously with the fermentation, which are incorporated without any major change into the molecule of the fermentation product, and which generally serves to increase the yield or improve the quality of the product. Okay, so precursors, they have two major roles to be played, that is it should be simultaneously added and it should get incorporated without causing any major change in the product or the molecule. Okay, and secondly, it should increase the yield and improve the quality of your product. Okay, so they are required in certain industrial fermentations and are provided through crude nutritive constituents, that is corn strip liquor, or by direct addition of most or more pure compound. Okay, so here are some examples. You can see the list here of precursors for some products, like you can see in case of penicillin G production for microorganism like Penicillium chrysogenum, phenylacetic acid is added. Okay, then in case of vitamin B12 production by Propionibacterium and Streptomyces species, cyanides are to be added. So they are the precursors and they are to be added in your media, but they will not harm your culture and the molecule or the product that is being synthesized. Okay, so these are the precursors. Then comes the inducers and elicitors. So if product formation is dependent upon presence of a specific inducer compound or a structural analog, it must be incorporated into the culture media or added at a specific point during fermentation. Okay, so if the process is dependent on a specific inducer compound or a structural analog, then it is to be added in your culture media. Okay, so that you get your desired product synthesized. The majority of enzymes of industrial interest, they are inducible. So inducers are often substrates such as starches and dextrins for amylase. This is one of the example. And in plant cell cultures, the in plant cell culture, the production of secondary metabolites such as flavonoids and terpenoids, they can be triggered by adding a elicitor. Okay, so this is the list here. You can see the inducers, they are given here. For example, in case of alpha amylase, inducer used is starch and maltose, and the organism or the microorganism that can be used is aspergillus and bacillus cetullus. Okay, then in case of cellulase, you can see cellulose is the inducer and trichoderma viridans is the culture that is used. Okay, so in case, so how it goes, so suppose this is the culture that you are using for a particular product. So the inducer for amylase is the enzyme which degrades the starch. So starch should be present in your media so that the amylase enzyme can degrade it, can work it, and can give out the product. Okay, that's how it works. Then comes the inhibitors. Now inhibitors are used to redirect metabolism towards the target product. Okay, it redirects the metabolism towards the target product and reduce formation of other metabolic intermediates. Other half of the pathway at a certain point to others, they halt the pathway at a certain point to prevent further metabolism of a target product. That is also a role of inhibitor. An example of inhibitor specifically employed to redirect metabolism is sodium bisulfite. Okay, so inhibitors, they will inhibit the process. Okay, you can see here it is given that they halt a pathway at a certain point to prevent further metabolism. So inhibition of a process is done so that further metabolism of a target produce is prevented. Okay, so this is the list here. So in case of, you can say glycerol, if it is a product, then sodium bisulfate is used as inhibitor, and it mainly affects the acetaldehyde production. Okay, that is repressed. And microorganism that is used here is Saccharomyces cerevisiae, yeast culture. Okay, so this is how you need to study the inhibitors and whether you are required or your media requires this or not. Then comes water. So all fermentation processes except solid state fermentation, SSF is solid state fermentation, requires vast quantities of water. Not only is water a major component of all media, but it is important for ancillary services like heating, cooling, cleaning, and rinsing. So these are the um, steps which are followed during fermentation process. You need to heat the fermenter to maintain the temperature. You need to cool it again if the temperature is too high. Then while cleaning and rinsing also you need water. So overall in fermentation process, water plays a very important role. Then a reliable source of large quantities of clean water of consistent composition is also essential. Then assessing the suitability of water like the pH, dissolved salts, then effluent contamination, the reuse of water is also important. It reduces the water cost by 50% and effluent treatment cost by 10 folds. Okay, so how you assess the suitability of water? You study the dissolved salts and pH, whether there are some effluent contaminants present or not, and you have to focus on the reuse of water. Okay, why? Because it reduces the cost by 50% and the effluent treatment cost, okay, that is the wastewater treatment cost, by 10 folds. Okay, then comes the oxygen, very important part if you are doing fermentation by using a aerobic um, culture. Okay, so for example, in case of cell biomass production, so where you, for example, say for um, fertilizer, where you need a large amount of cell biomass as a product to be used further, you do fermentation. So in that case, aerobic culture comes into picture, so it requires oxygen. So depending on the amount of oxygen required required by microorganism, it may be supplied in the form of air containing about 21%. volume by volume of oxygen, or occasionally as pure oxygen when requirements are particularly high. Now you study oxygen as dissolved oxygen as well. Okay, so depending on that, you decide whether you need to supplement or supply the oxygen or not. The organism's oxygen requirement may vary widely depending on the carbon source. For most fermentations, the air or the oxygen supply is filter sterilized prior to being injected further into the fermenter to avoid the contamination. Okay, so this, so last point from today's video, that is antifoam. Okay, so foam is something that is formed during fermentation, and antifoams are necessary to control the foam. So foaming is largely due to the media that contains proteins that becomes attached to the air broth interface, and where they denature to form a stable foam scheme that is not easily disrupted. Okay, so an ideal antifoam agent is used, and it should have following properties. It should disperse readily and have fast action, active at low concentrations. That means you don't need to add 100 ml, 200 ml, because it will also increase the volume of your culture media, and it can dilute your culture media. Okay, so very low, even it should be active at very low concentrations. That is very important. It should be long acting in preventing new foam and should not be metabolized by the culture. So it should not be toxic to microbes. That means it should not control, or it should not reduce the bacterial growth rate, and it should not harm your product. Okay, so it should not be toxic to microbes and humans, etc. It should be cheap and should not cause any problems in fermentation process. Okay, so this was all about the fermentation media. So I hope I have covered everything, every point here. So I hope you like my video. Do give a thumbs up, do share my videos with your friends, and do subscribe to my channel and hit the bell icon so that you will get notified on each new video that I upload. Thank you.